Hello everyone, welcome to today's video. In this video, we'll be understanding quorum sensing in Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative bacteria which is known to be present in our environment. It is an opportunistic pathogen that can cause various kinds of infections in humans, animals, etc. And that's why it becomes very, very important to understand quorum sensing signaling system in Pseudomonas aeruginosa, because this regulates the virulence in Pseudomonas aeruginosa. Right? There are different quorum sensing systems in Pseudomonas aeruginosa. There are at present 4 quorum sensing systems that are known: LasA system, AHL system, you have PQS system, and you have IQS system. Every system will have a fundamental, building blocks that building blocks that construct the system. So we'll understand LasA system first, because LasA system is the master regulator of virulence gene expression, and it also regulates the other quorum sensing pathways. So that's very, very important for us to start with the LasA system, right? So we'll start with the LasA system. First, I need to create the cell, because inside that cell we'll be creating our further illustrations. And as you can see, what we have is the bacterial cell, gram-negative bacterial cell. You have surface structures, you have flagella also. So this is what I've already created, and inside that we'll try to understand the LasA system. So the LasA system overview, if we try to understand the overview of LasA system, it is a cell density dependent communication system that allows the bacteria, which is Pseudomonas aeruginosa, to sense the population. So what happens is, bacteria, as we know, bacteria is a single celled organism, and many times what people think is it's a very, very boring kind of organism which actually grows and then divides and die. But there are regulatory processes that happens inside Pseudomonas aeruginosa and other bacteria also, which are example is quorum sensing system. So it's cell density dependent system means when the bacteria is going to grow and you have more bacteria, you have a biofilm of bacteria, this system is going to get activated, and that will control the virulence expression of virulence genes. So at low density, what happens is it secretes a lot of signal molecules. Those signal molecules those are required for this signaling pathway. So this is what is the representation for these signaling molecules. So let's start from here, that bacteria is producing few signal molecules, and each bacteria will have a capability to produce certain amount of these signal molecules. These signal molecules will go out like this, and if you have one bacteria, you'll have, let's say, 3 signal molecule outside. If you have multiple bacteria, you'll have multiple signaling molecule out, right? And these molecules also have the capability to come inside the cell, and in that sense, bacteria is able to detect how many signaling molecules that are there in the environment. Right? Because bacteria, they do not have any sensory organs like we have eyes, so that they can see how many bacterial cells are there in the environment. So what they have is a chemical signaling system which helps them to recognize how many cells are there in the environment. And if you have more cells, that means you have more power, and you can basically target the host and maybe occupy that particular environment. So it's a strategy of the bacteria that it uses against the host, right? So these are the signaling system. These are the signaling molecules that are produced by a particular enzyme called LasI, this particular enzyme. So let me bring that enzyme inside the cell. So this particular enzyme, which is LasI, will produce a lot of these kind of signaling molecules, and these signaling molecules, they will be produced from a particular substrate. Because I want to also talk about how these signaling molecules, they are produced. But before that, let's understand what are these molecules. These signaling molecules, they are called AHLs. AHLs are acyl-homoserine lactone molecule. For LasA system that we are understanding over here, the molecule is this particular molecule, right? Okay, so let me copy this and then zoom in a little bit so that you can see the structure. So it's called acyl-homoserine lactone molecule. In case of LasA system, you have 3-oxo C12 homoserine lactone molecule, and this is the fundamental, structural component of that molecule. You can see you have homoserine lactone ring that is common in case of all the AHL molecules. AHL molecules are having different carbon chain. That R group that you're seeing is basically the length of it's R group, which is having the different length of the carbon chains. So you'll have C12, you can have C6, you can have C8, you can have C4. C4 is the ligand for RHL system. As I already told you that in case of Pseudomonas aeruginosa, you have multiple systems. The second one, widely studied system, is RHL system. In that case, you have RHLI as the inducer, which creates that particular signaling molecule, which is C12 homoserine lactone molecule. In case of this particular system, which is LasR system, you have 3-oxo C12 homoserine lactone. So that means you will have like total 13 chains, 13 carbon molecules like that. So it will have long chain over here. So basically what you have is one part which is homoserine lactone molecule. The second part is the fatty acid acyl chain. And because of this fatty acid acyl chain, the molecule can move around. It can go outside. It can come inside the bacterial cell very, very easily because of the hydrophobic nature of this particular molecule, right? So that was all about the signaling molecule. Let's start with the LasI protein. It's also known as auto-inducer synthase. So I for inducer. So LasI, I for inducer, LasR, R for receptor. I'll come to LasR after I'll explain the auto-inducer synthase. So auto-inducer synthase, this particular synthase is an enzyme. It works as an enzyme which produces this particular signaling molecule, which is 3-oxo C12 homoserine lactone molecule. And it is constructed from different substrates. There are two important substrates that are required to produce these molecules. And I want you to take notes if you basically want to understand the synthesis of AHL. You have SAM, which is S-adenosylmethionine. This is SAM, and then you have acyl carrier protein, which is acyl-ACP. Both of these work together to create this with the help of our LasI protein. So LasI protein is also very, very important target when it comes to inhibition of the quorum sensing signaling pathway. Right? Okay, so this is going to produce this acyl-homoserine lactone molecule, and this molecule will simply diffuse into the environment like that, right? So for that, probably I think let's use an arrow to showcase that this molecule is being produced by this particular protein, which is LasI over here. Now this will create the molecules, and they will freely diffuse out into this surrounding, and that is the continuous supply provided by the LasI. Now what happens is these molecules, as I told you, that after having a particular threshold concentration of these molecules in the environment, because this will happen, if you have lot of molecules coming from different bacteria, they can also come back inside because you will have high concentration to low concentration of those molecules. And then what will happen, these molecules will come inside the cell, and there is one protein that is called LasR protein. It's called master regulator also. It's called transcriptional regulator also. This can bind with this particular receptor like this. It will have two domains basically. This LasR usually have one is ligand-binding domain. Ligand-binding domain means ligand, that is this acyl-homoserine lactone molecule, that is able to bind to this particular domain, and it will have the DNA binding domain. The second domain is the DNA binding domain. And from that side, it can bind to your DNA molecule. So it becomes the transcription regulator, right? So what happens after binding, it changes its conformation and then move and bind to the DNA molecule. So here is DNA, which I've already created so that you can understand. Now this is the DNA. What happens is this protein is going to move and it's going to bind to a particular region called LAS box. That LAS box is a region, it's a promoter region. LAS box specific sequence, it's a region, DNA region, where this particular protein goes and bind, and it will bind like this. It will have a particular site, especially in the second domain, that will help it attach to that particular region like that. And then what it does, it starts the expression of various genes that are under the regulation of LAS system. Right? So the binding of the molecule that is coming from the outside is going to stabilize this protein. Also, usually it's a very, very unstable protein. So when the molecule comes inside, it stabilizes and creates the complex or make the complex, which is active LAS and AHL complex. And this activated LAS complex bind to the specific promoter region, which is called LAS box sequence, and it will initiate the expression of various proteins, various valence factors. In this case, you also have another important regulator, regulator. It's also known as negative regulator. So I've changed this color into a little bit darker than the LasR color. As you can see over here, this is another regulator. It's a negative regulator. So this particular molecule, that negative regulator, is involved in inhibition of the LasI expression. So it's going to stop the expression or reduce the expression of LasI protein, so that there is no excessive production of these molecules from a single bacteria. So it's a kind of a feedback loop, that how many molecules are being produced in the environment, and that should activate further. The concentration of these molecules should activate this also, because this is going to further then tell the RHLI system to then stop producing, because now it's enough AHL in the environment, something like that, right? So you have a kind of a feedback loop that will control the expression of these molecules. Now, what this interaction is going to do, it's going to produce important virulence factors, starting from one alkaline protease. This is one virulence factor. So I'll show you that the production is coming from this interaction right over here. So this is producing here, the downstream of this particular region of the DNA, you have these particular genes. Second one is LasA protease, right? The first one is alkaline protease. Second one is LasA protease, and then LasB elastase. These are the three important virulence factors that causes a lot of tissue damage in humans when there is infection because of the Pseudomonas aeruginosa. As I told you, it's opportunistic pathogen when immunity is low, or there are some other complications associated with the human body. This bacteria can cause infection. Okay, such as in case of cystic fibrosis, you have infection in the lung because of Pseudomonas aeruginosa, and it can cause a lot of damage in the lung because of these virulence factors, right? There is another component which is called efflux pump. Let me discuss the role of efflux pumps over here also. These efflux pumps, they are present in the membrane like that. So I can show a couple of efflux pumps right over here also. The role of these efflux pumps is actually they can export out these molecules if they are present in very high concentration. So these can export these AHL molecules out against the concentration gradient. So indirectly they can also control the concentration of these molecules inside the cell, outside the environment, how many molecules should go out, right? So that is also been controlled by these efflux pumps. And these pumps, they are also very, very important in case of antibiotic resistance, because if antibiotic comes inside like this, they can be thrown out by using efflux pumps. So efflux pumps are really important. That regulates the concentration of these molecules inside as well as outside of the environment in a way that is required by the cell. The next one is virulence factors. So we'll talk about the first virulence factor, which is alkaline protease. Since it is a protease, so it's going to degrade protein. So that's why it's very, very important in degrading. This particular protein is important in degrading cytokines. I'm sure that you know that our immune system produces a lot of cytokines, and these cytokines can cause activation of various cells, and then that is a problematic situation for our bacteria, because it will not survive if you have a lot of cytokines attracting a lot of immune cells. So it try to act on the cytokines so that it can reduce the expression of the cytokines, and there is less activation of immune cells. Next is it can also inhibit the immune signaling and phagocytosis. When phagocytosis is happening, it can actually cleave a lot of proteins that are required for that signaling, and it can help bacteria survive during infection. Next you have is LasA protease. LasA protease right over here that I've shown you. LasA protease is a particular protease that is very, very important when it comes to degradation of the epithelial barriers. Bacteria need to penetrate into various organs, right? So you need to break that epithelial barrier. For that, LasA protease is very, very important. So as you can see, now the bacteria started producing these molecules. So it's a direct attack on the host. So with the signaling molecule, because now bacteria is sure that it has a lot of different other bacteria, as its army is huge, so it can attack on the host system. So it start producing all of them, not only one bacteria, all of them will start doing the same thing, and it will cause a lot of damage to the host. On the other hand, when the cell is not activated, the system is not activated, cells, they are there in a low concentration or low amount, it will not be activated. So it will just stay quiet. So the immune system is not working against this bacteria also. So it tried to be in a dormant condition, let's say, right? So third virulence factor is your elastase. Elastase is again highly potent virulence factor of Pseudomonas aeruginosa that can degrade elastin, collagen, immunoglobulins, antibodies, right? It can even degrade antibodies. It can degrade collagen, which is highly resistant protein. It can degrade elastin, which is very, very important protein. So you can imagine how much damage it can cause, especially in case of lungs. A lot of damage is caused by this particular enzyme. So if you try to develop therapeutic molecules against this particular protein, definitely there will be a lot of damage that will be reduced in case of cystic fibrosis patients if we can counteract on this particular protein. Now it's also associated. And not one single protein, but all of them are associated with that, lot of tissue damage and tissue necrosis. So that is why it's very, very important to understand the signaling pathway. What are the molecules that are involved in Pseudomonas aeruginosa signaling and how it works? What are the targets for drugs that you want to find out? And that reminds me to actually tell you that about my resource that I've created, because in that resource we have lot of amazing courses that you can do and do great drug discovery research. Let me show you that. This is the resource that we have created called ResearchGoal. You can just type researchgoal.com and you will be directed to this particular website where we have multiple different courses that I've created. And let me show you these courses right over here. There are courses that you can complete, courses related to drug discovery, courses related to bioinformatics. There are various courses that I've created and the price is not that much high. You get a lot of high quality content. You'll get video content like this, and I'll take you through all these complex topics, and you will be able to perform these kind of experiments very, very easily. All these are little bit advanced courses. If you're interested in research, you should be doing those courses. For example, I have a course on molecular docking. If you want to understand molecular docking, this is the course. Right over here, if you go down in the website, I've already mentioned right over here that you have drug discovery learning pathway. So you can start with this particular course, and then second you can do molecular visualization, then you do protein editing, and then you molecular docking. You will get all the skill sets that you require for the drug discovery using molecular docking. Because after that, I'm also planning to have molecular dynamic simulation. We'll see how that goes, right? And second one is biological data analysis. If you want to do rigorous data analysis, you can do Python-based data analysis. You can do first you need to understand Python, then you do Python-based data analysis, then you do R and R foundations, and so on. So this is the resource that I've created, and you have contact us tab over here. Detail is also mentioned. My contact number is also there. You can direct message me. You can direct email me. If you want to type your query over here directly, there is no need for you to open email, and you can just send me the message. I'll get back to you as soon as possible, right? So please contact me, and if you have any doubt, if you have any query, please let me know and do these courses. Let me tell you, these courses are coming from my own experience. I'm creating those courses. Why? Because I face a lot of challenges. My students are facing lot of challenges. So I've created these courses so that research becomes much smoother, and I'll be bringing a lot of amazing courses also in this particular resource. Once you take the course, the entire update in that particular course will be free to you. You can have that update anytime you want, and if you have that skill set, you should be able to publish high impact reviews, and without any lab facility, you can become researcher. You can have impact on the science. That is very, very important, right? So I request you to get enrolled to these courses, and let me know if you have any doubt or questions regarding any of that. All right, I hope you will do some of the courses, and I'm really looking forward for your support to the resource. And if you have any doubt, any questions, you can put it in the comment section, and you can also contact us directly. All right, so as you can see over here, this is a very complex system, and when we talk about biological significance of this particular system, Pseudomonas aeruginosa LAS system is the master switch for pathogenic behavior of Pseudomonas aeruginosa, because it can control various virulence factors, determinants. At the same time, it can also regulate the population, right? So that is why this has been a target for discovering various drugs that can reduce the virulence of this particular bacteria by targeting this system. What will happen if we target this system? Then expression of the virulence factors will not happen, and the tissue damage will be reduced. So you can say it's a new kind of antimicrobial therapy where we are not killing the bacteria, but we are suppressing the virulence. That's why this therapy is also known as virulence therapy. So take-home message is basically the central regulatory hub of signaling system in Pseudomonas aeruginosa is LasA system, and you'll have multiple components. You have LasI right over here. You have LasR. You have RHL. Sorry, not RHL, but RSAL, and then you have efflux pumps. You also have acyl-homoserine lactone molecules that control this entire system, and because of this, Pseudomonas is able to control the host and survive under the difficult conditions. And this is very, very important when it comes to development of next generation antimicrobial strategies, because you can target this system, and this can be wonderful topic for your research, and you can come up with amazing molecules, and you can test these molecules. And it's also been tested in clinical trials that these molecules have some promise against the infections caused by Pseudomonas aeruginosa, especially when we don't have antibiotics against the bacteria, because many of the bacteria, they are antibiotic resistant. So we need to have new molecules, we need to have new therapy, and we can target the systems like this, as we've seen in Pseudomonas aeruginosa, in this bacteria, in other bacteria also, so that we can develop new therapies that will help us take care of these dangerous bugs, and hopefully will help in developing new kind of therapeutics, right? All right, thank you so much for watching the full video. I'll meet you in the next one where I bring similar kind of topics, and if you have any suggestions, please feel free to send me the message, post the comment, and I'll get back to you as soon as possible. Thank you and take care.